Welcome to your final web lecture of the semester. Now that we have reviewed the basic principles of cell signaling and we've taken a look at some of the specific kinds of hormonal signaling pathways within animals, now it's time for story time. We're going to go through some specific examples of hormone pathways in animals, and we're going to focus in on pathways related to homeostasis, development, and behavior. We're going to start with the idea of calcium regulation. So, as we know, calcium ions are extremely important to several processes that we've seen, and also very important to several processes that we haven't looked at in detail. So we saw that calcium plays the critical role in the release of neurotransmitter in the axon terminals of neurons. We've seen its use as a second messenger. What we didn't get to is its role in muscle contraction, but it turns out that calcium ions play a central role in the ability of muscle cells to shorten. So calcium levels in the body are extremely important. In mammals, the levels of calcium in the system are partially regulated by a hormone called parathyroid hormone. Parathyroid hormone is released by the parathyroid glands. They're actually embedded, these tiny little glands embedded in the posterior surface, so the back surface of the thyroid gland there in the neck. And this is activated when calcium levels fall below a particular set point. Parathyroid hormone works to increase the level of blood calcium ions when they fall too low, and it's going to do this in a number of ways. So if we look at this negative feedback regulation chart, the parathyroid glands are going to secrete parathyroid hormone. One of the effects is going to be to increase the reabsorption of calcium ions from the kidneys to try to increase the circulating levels, make sure that not too much of it is excreted through the kidneys. And it's also going to mobilize calcium ions from the bones. So let's take a look at how it does that. So this is a diagram of normal bone remodeling and repair. And the first thing that happens is these cells called osteoclasts. So osteoclasts are bone dissolving cells. They're going to secrete a strong acid along with enzymes that break down the collagen and other fibrous materials within the bone. And it's just going to create these tunnels through the bone as it moves through. So here we're seeing an earlier point in time, later and later and later in time as these osteoclasts tunnel through. So this is earlier where the osteoclasts have been working and there's this hollowed out tunnel going through. And then normally what happens is that there are another population of cells called osteoblasts that come and line the interior of that tunnel that the osteoclasts have created, and their job is going to be normally to lay down more bone material from the inside of that tunnel to regenerate the bone. And so this is what a cross-section through that would look like later on when the osteoblasts have laid down a bunch more bone. So one of the things that parathyroid hormone does is it activates these osteoclasts in the bone to release a bunch of that calcium into the blood. And of course, if calcium levels continue to be low, that's not going to be replaced by these osteoblasts. We're just going to be able to use that calcium. So this enables the skeletal system to be an important reservoir for physiological calcium that's needed to perform all these really important functions that calcium ions do. And the idea is eventually you get more dietary calcium in and you're able to replace that bone using these osteoblasts. It also indirectly affects calcium ion uptake by promoting the production of vitamin D. So vitamin D is going to be synthesized in an inactive form in the skin cells by exposure to sunlight and in some other ways, and parathyroid hormone is going to activate that vitamin D. Vitamin D makes it easier for the small intestine to absorb calcium ions. So this is why we have vitamin D in milk, because when it accompanies the calcium, it makes that calcium absorbed more readily. So this is going to increase the circulating calcium ions in the blood. A negative feedback loop is going to tell the parathyroid glands to stop releasing parathyroid hormone, and balance will be attained. There's another factor, though, that also plays the opposite role in this whole process, and that is the hormone calcitonin. So calcitonin is released by the thyroid gland, and it is going to work to decrease the level of circulating calcium ions in the blood. So if there's too much calcium in the system, it can cause little mineral deposits in the body that are not very good for you. And so the body also doesn't want to let calcium levels get too high. So what is it going to do? It's going to do pretty much the opposite of what parathyroid hormone does. It's going to increase the excretion of calcium by the kidneys, so it's going to inhibit the reabsorption of calcium ions in the kidney, and it's going to inhibit osteoclasts and stimulate osteoblasts. So it's going to inhibit those bone dissolving cells and stimulate those bone secreting cells to store more of that calcium in the skeleton, so that the skeleton can continue to be used both as a support system for the vertebrate body and also as a reservoir of calcium for physiological needs. So again, we're going to stimulate these little cells that are lining the tunnels that were made by the osteoclasts to replace that bone that was lost when the osteoclasts tunneled through. So these two work together to make sure that those calcium levels stay just right. So you can think of this as temperature control within a building that has both a furnace and air conditioning that's going to be making sure that the temperature in that building stays just right by cooling it if it gets even a little bit too warm, warming it if it gets a little bit too cold. And that is regulating it from both directions is going to keep the level absolute steadiest. So we've got calcitonin, making sure the levels don't get too high, and then parathyroid hormone, making sure the levels don't get too low. Now let's take another look at antidiuretic hormone, a more detailed look at it. We saw this before when we were looking at water balance, when we're thinking in general about homeostasis in animals. Now let's take a closer look at exactly how this happens. So we saw that this response mostly has to do with the release of ADH, antidiuretic hormone, which is going to have a number of effects. One of them is reducing the urine volume. Another one is increasing vasoconstriction to help to increase the blood pressure while the body is getting rehydrated. We also have seen in the last web lecture that antidiuretic hormone is one of the hormones produced by the posterior pituitary, which remember is an extension of the hypothalamus. These neurons, these neurosecretory cells in the hypothalamus that produce the antidiuretic hormone, the hormone is actually going to travel down the axons to the axon terminal where these neurohormones are going to be secreted and picked up by the blood vessels to travel around to their target. Let's now take a closer look at what happens within the kidney to help to reduce the urine volume. So I'm going to go very briefly over the structure of the kidney, just enough for you to understand how this works. So the kidney consists of a series of these little tubules that carry fluid that's been filtered from the blood. So the blood is going to be forced through these little capsules into this tubule system. So the fluid and the small molecules are going to be in this tubule, and this is a mechanism for sort of cleaning the blood, getting rid of metabolic wastes and other things that are in the blood, and it's also going to play a role in water balance. So as this filtered fluid, this filtrate we call it, comes into the tubule, some materials that the body needs to retain are taken back up into blood vessels, and the filtrate moves through the system of tubules. So in birds and mammals, the tubules include these long loops called loops of Henle, and the purpose of these long loops is to generate a solute gradient from the external part, so this is the outside cortex of the kidney, down into the inner, what we call the medulla of the kidney. So as you go down through this tubule, it's becoming saltier and saltier and saltier, and don't worry about the details of how this happens, but it's actually the movement of this fluid through this tubule that creates that salt gradient. So as it goes through the tubule, it's going to come to this final section called the collecting duct, and this is where the water balance regulation actually occurs. So as it goes through this collecting duct and descends down through this solute gradient, water is going to be taken back up out of this filtrate. It's going to diffuse out more and more as it gets into the more and more concentrated areas of the inner medulla. And the permeability of the walls of the duct is going to determine how much water is retained in the urine and how much is absorbed back into the blood. So this is going to be surrounded by blood vessels, and so water that moves out is going to move into the surrounding blood vessels. So now let's take a look at how that works down at the cellular level. So what we're looking at here is a cell in the wall of that collecting duct. So this inside that's colored appropriately yellow, that's where the urine is being created. This is the inside of that tubule. The other side is the interstitial fluid outside that collecting duct, and then a nearby blood vessel is here. When antidiuretic hormone is released, it's going to move out of the blood vessel and it's going to bind with its receptor on the outside of this collecting duct cell, and it's going to be a G-coupled receptor protein. And when that G protein is activated, it's going to activate adenylyl cyclase to synthesize cyclic AMP from ATP. So we've got a second messenger being synthesized. Cyclic AMP activates protein kinase A to mobilize motor proteins that are going to deliver these little vacuoles with aquaporin channel proteins embedded in the membrane out to the cell membrane. It's going to fuse with the cell membrane and insert those aquaporin proteins into the cell membrane. So remember that what aquaporins do is they don't change the osmotic gradient. They don't change which direction the water is going to flow, but they increase the rate of osmosis. The more aquaporins you have, the greater the rate of osmosis along that water potential gradient. So aquaporins increase the rate of osmosis from the filtrate, this forming urine within the tubule, which has a less negative solute potential. Remember, we're in the most concentrated part of the medulla of the kidney. So this is going to have the least negative solute potential, the lowest osmolarity. So we see the osmolarity of around 300. So water is going to flow from this less concentrated filtrate into the cell. There are also going to be aquaporins inserted onto the outside of these cells, out facing the interstitial fluid. Water is going to flow from the inside of the cell to this very concentrated region out here in the renal medulla. We have a concentration of about 600 milliosmolar here, and then finally it's going to diffuse by osmosis into the bloodstream, which has a solute concentration of about 700. So it's going to follow that solute gradient, that solute potential gradient from the urine in the tubule all the way out to the bloodstream. If there are a lot of aquaporins there, that's going to be the direction of flow of water. When there are fewer aquaporins, it's going to do this at a lower rate. So when ADH is not present, there are fewer aquaporins. There's going to be less water flow from this collecting duct. So the urine will be more dilute. It'll have more water in it. When antidiuretic hormone is present, there's going to be a lot of flow through to the bloodstream, and this urine will be more concentrated. So ADH has other functions in animal bodies aside from water regulation. It turns out that antidiuretic hormone has another name. It's also known as vasopressin. It was given this other name because it has other functions, and it wasn't discovered until later that it was actually the same hormone, the same molecule that's doing both things. And so ADH and also oxytocin work together to affect social behavior in different ways. So some of the experiments were done on these species of voles. There's the prairie vole that has this sort of unusual behavior among voles for forming very solid pair bonds and also having very devoted, intensive parental care. And so these prairie voles were compared to the more solitary meadow voles that do not form these pair bonds and have very little parental care. Researchers looked at the activity of vasopressin, or ADH, in the brains of these different kinds of voles. And what they found was that there was high vasopressin activity, ADH activity in the prairie voles normally, and very low in the meadow vole. So they did a manipulation experiment where they added a synthetic form of vasopressin, injected it into the meadow voles, and found that this significantly increased the vasopressin activity within the brain. So that indicates that the vasopressin receptors are there in the meadow voles. They just don't produce as much vasopressin as the prairie voles, and this was also reflected in the social behavior of these three groups. So prairie voles spent much more time in association with their partner than they did other voles that they didn't know. Meadow voles spent more or less the same amount of time with individuals that they'd mated with as opposed to individuals that they didn't know that they hadn't mated with. But then when they were injected with this synthetic vasopressin, that percentage increase. They ended up spending more time in association with a mated partner than individuals that they didn't know. A similar kind of comparison was made between the prairie vole and another subspecies, closely related subspecies of vole, the montane vole, that is also known for solitary behavior. And it looked at the number of oxytocin receptors in the brain. So oxytocin is another hormone that's known to influence social behavior. And it was found that the prairie voles had a lot of receptors for oxytocin in their brains. The montane voles had very few. And then if we looked at the vasopressin receptors, we see a large number of them in this region of the brain and very few in the montane voles. So we can see that these two hormones, oxytocin and vasopressin, often referred to as the love hormones, both of these play a role in pair bonding and parental care. Now let's take a look at stress responses and the adrenal hormones. So the adrenal glands are located on top of the kidneys. So they are these little masses of glandular tissue that sits right on top of the kidneys. And each adrenal gland consists of two separate but fused glands. The adrenal medulla, which is the inner portion, and the adrenal cortex, which is the outer portion. These different portions have different developmental pathways. So the inner portion, the medulla, is derived from neural tissue. So this is the neural part of the adrenal gland. So the adrenal cortex is derived from glandular epithelium, and that's sort of the regular gland part of the adrenal gland. So let's first take a look at the role of the adrenal medulla, this interior part that's derived from neural tissue. So the adrenal medulla secretes two hormones, epinephrine and norepinephrine, also known as adrenaline and noradrenaline. Both of these hormones also are used throughout the nervous system as neurotransmitters. And these hormones are members of a class of compounds called catecholamines. And catecholamines coordinate a set of physiological responses that comprise the fight or flight response. So they're going to mobilize energy resources to the skeletal muscles to prepare for a physical response to some kind of dangerous situation. So some of the effects of epinephrine and norepinephrine is triggering the release of glucose and fatty acids into the blood to be used as a ready energy source, increasing oxygen delivery to body cells to increase the rate of aerobic metabolism to be ready for a high level of physical activity, also directing blood toward the heart, brain, and skeletal muscles. These are going to be the things that are really important in an emergency, and away from things like the skin, the digestive system, kidneys, reproductive system, things that can wait until the emergency is over. So, as we just saw, epinephrine has different effects on different kinds of body tissue. So let's take a closer look at some of the ways that that happens. So, as we've seen, epinephrine coordinates a response in a range of different kinds of target cells. In the last web lecture, we saw that there are two classes of receptor cells for epinephrine, alpha and beta. In liver cells, it's going to bind to a beta receptor that's going to activate a protein kinase A, which regulates glycogen metabolism. We've already looked at this pathway in detail, particularly in how that signal is amplified to create a lot of glucose to be released into the blood from the liver. In smooth muscle cells that line blood vessels that supply skeletal muscle, so these are going to be supplying blood to get you ready for some intense physical activities. It's going to lead to vasodilation to increase the blood supply to those skeletal muscles. So again, it's going to be received by a beta receptor, and it's going to go through some pathway that's going to lead these cells to relax. Remember, muscle cells always shorten when they contract, so relaxing those cells is going to increase the diameter of those capillaries and allow more blood to flow through to those skeletal muscles. So the blood vessel dilates, it becomes larger in diameter, increasing the blood flow to the skeletal muscle. But if we look at smooth muscle cells of blood vessels that go to the intestines, so remember, we're going to decrease our investment in digestion and other activities that can wait until the emergency is over, so we want to restrict the blood flow to those organs. So in a smooth muscle cell in the wall of a blood vessel that supplies the intestines, we're going to have a different kind of receptor. It's going to be the alpha receptor that's going to interact with epinephrine. So it's the same kind of cell, but it's going to have a different response. When that alpha receptor is activated, the cell is going to contract and shorten, and that's going to shrink the diameter of those blood vessels and allow less blood to flow through. So we see in the case of the liver cell and the smooth muscle cell going to skeletal muscle, the same receptor, but there are going to be different intracellular proteins that are going to lead to different kinds of responses in these very different kinds of cells. Here we have the same kind of cell that's going to have the same basic kind of machinery, and we have different receptors that are going to cause different responses in these very similar cells. So now let's take a look at the role of the adrenal cortex in this whole stress response system. So the adrenal cortex is also going to respond to stress, but not so much the imminent danger kind of stress. These are going to be generally stressful conditions related to deviations from homeostasis, so including low blood sugar, decreased blood volume and pressure, and shock, things like that, that are basically not immediately life-threatening but need to be brought back into balance. So a series of hormonal signals lead to the production and secretion of a family of steroids called corticosteroids. And humans produce two different types of corticosteroids, glucocorticoids and mineralocorticoids. Glucocorticoids, such as cortisol, influence glucose metabolism and the immune system. So they're going to not necessarily cause the release of glucose, but increase the use of glucose by the cells and the delivery of glucose to the cells. Mineralocorticoids, such as aldosterone, affect salt and water balance. So let's take a look at the big picture of the coordination between the nervous system and endocrine system in these adrenal glands. So remember that the adrenal medulla is more closely aligned with the nervous system. It's derived from nervous tissue. So this is going to be under the control of the sympathetic nervous system. So coming from the central nervous system, there's going to be a pathway, a neuronal pathway, that's going to lead directly into the adrenal medulla to activate those epinephrine and norepinephrine release, leading to glycogen being broken down into glucose, increasing the blood glucose, increasing the blood pressure, so delivering blood at a greater rate to the muscle tissues, increasing the breathing rate to increase the oxygen supply, increasing the metabolic rate, mobilizing energy, and change in blood flow patterns, as we saw before, leading to increased alertness and decreased digestive, excretory, and reproductive system activity. If we think about the other side of this, the more endocrine side, so remember that the adrenal cortex is derived from glandular epithelium. It's going to be activated in a different way. The hypothalamus is going to release ACTH releasing hormone to the anterior pituitary. So the adrenocorticotropic hormone that's going to be released from the anterior pituitary is then going to be released to the circulation, go around to all the body tissues. The ones that arrive at the adrenal cortex are going to stimulate the production of these mineralocorticoids and glucocorticoids. So the mineralocorticoids are going to influence the retention of sodium ions and water by the kidneys and increase the blood volume and blood pressure. The effects of the glucocorticoids are going to be that proteins and fats are broken down. So if the blood sugar is low, that's telling the body that not enough food is being consumed. The body's going to start breaking down its proteins and fats to create more glucose. And so these are going to be converted to glucose, leading to increased blood glucose, and then also partially suppress the immune system to conserve that energy that would be used by the immune system to be used for more basic body functions if there's some sort of food emergency. Now let's turn our attention to the sex hormones. So the gonads are the testes and ovaries, and they are going to produce most of the sex hormones. And the sex hormones are classified as androgens, estrogens, and progesterone. All three of these sex hormones are found in both males and females, but in different proportions. So the testes primarily synthesize androgens, mainly testosterone, which promote the development of male reproductive structures. And testosterone is also responsible for male secondary sex characteristics, so all of the things that make males different from females in non-reproductive ways. Estrogens, and most importantly estradiol, are responsible for maintenance of the female reproductive system. And they're also responsible for development of female secondary sex characteristics, so all of the things not related to reproduction that are different between males and females. In mammals, progesterone is primarily involved in preparing and maintaining the uterus, so it's going to be a regulator of menstrual cycles and have a big role in pregnancy and childbirth. Synthesis of the sex hormones is controlled by the gonadotropins, So these are going to be released by the anterior pituitary. The two gonadotropins are follicle-stimulating hormone and luteinizing hormone. So these are going to be under the control of the hypothalamus, and the releasing factors from the hypothalamus, so a signal from the hypothalamus, will tell the anterior pituitary to release these gonadotropins. Gonadotropin secretion is controlled by gonadotropin-releasing hormone from the hypothalamus. There have also been several substances discovered that work as endocrine disruptors, so they're going to tinker around with this endocrine system, sometimes in very unpredictable ways. So between 1938 and 1971, some pregnant women at risk for complications in their pregnancy were prescribed a synthetic estrogen. It's called diethylstilbestrol, or we'll just call it DES. So it turned out that the daughters of the women who were treated with DES are at higher risk for reproductive abnormalities, including miscarriage, structural differences in the reproductive system, and cervical and vaginal cancers. So this had its effect on that developing fetus while the mothers were taking this synthetic estrogen. It turns out that DES is an endocrine disruptor, a molecule that interrupts the normal function of hormone pathways, in this case that of estrogen. And so many of these have been discovered with both harmful and not so harmful effects. So we know that bisphenol A that's found in many softer plastics work in this way. There's been research that's shown that the average age of the onset of menstrual cycles in girls is getting lower and lower, so younger and younger girls are starting to cycle, and it's thought that this may be related to exposure to this bisphenol A in plastics. We've also discovered in recent decades that there are phytoestrogens, a form of endocrine disruptors in some plants, such as soybeans, that your book mentions can have some helpful effects in reducing the rates of breast cancer. So now let's think about these circadian rhythms, these biological rhythms, so circadian rhythms and seasonal rhythms. And these are generally regulated by a gland in the brain called the pineal gland. So it's located here, right in the center of the brain, and it's going to secrete a hormone called melatonin. So the primary functions of melatonin appear to relate to biological rhythms associated with reproduction and with daily activity levels. So reproduction in animals that reproduce seasonally, it's going to be the pineal gland that regulates behaviors based on the season. So these hormones are biological molecules that are going to be under the control of genes, and of course genes can mutate and evolve, and so these hormones can have different kinds of effects in different organisms as they have changed through evolutionary time. So over the course of evolution, the functions of particular hormones have diverged in different biological groups. For example, thyroid hormone plays a role in metabolism across many, many lineages. But in frogs in particular, it's taken on a unique function. It stimulates the resorption of the tadpole tail during metamorphosis. The cells in that tail undergo apoptosis, and those materials are reused by the body when the frog is losing that tail and growing legs. Prolactin also has a broad range of activities in different vertebrates, so the role that we have talked about is in stimulating milk production. Obviously, that's only going to be done in mammals. Mammals are the only vertebrates that produce milk, so prolactin is a hormone that exists in other vertebrate groups, but with different functions. Melanocyte-stimulating hormone, which is released by the anterior pituitary, regulates skin color in amphibians, fish, and reptiles by controlling the pigment distribution in melanocytes. So this is the way they can change color very quickly in response to the environment. But in mammals, MSH, the same hormone, plays a role in hunger and metabolism in addition to coloration. So this is a new function that it's taken on in this one taxonomic group. So I'd just like to wrap up this web lecture by sort of bringing the semester full circle and talking about what we said we were setting out to do at the very beginning of the semester, and a little bit about how we've accomplished many of these goals. So at the very beginning, I told you in the very first class that this is going to be a course about biological mechanisms. how does biological organization arise? Why do the different cellular parts, the different molecules in cells do what they do? What causes things? So we said in biology you'll hear lots of different explanations about molecules moving along concentration gradients, being attracted to opposite charges, having a natural affinity, binding reversibly to its receptor. And so we started out the semester by learning a lot about how these things come about, strong and weak binding, strong and weak attractions between molecules, covalent bonding, and how all of that comes about. And all of these molecular phenomena have been re-invoked again and again throughout the semester to explain how processes occur on larger scales. So we have these original themes that we started with at the beginning of the semester, one of which was that biological systems create order, create and maintain order. We saw how this happens seemingly against the law of thermodynamics, but what we saw is that biological systems can increase the amount of order, or rather decrease entropy at the local level, while still increasing the level of entropy overall between the system and its environment. We've also seen the mechanisms by which biological systems avoid reaching homeostasis to create greater levels of order. We've seen a lot about energy processing, both in the topics of photosynthesis and cellular respiration, all the way up to the organismal level, where we saw how energy is managed to regulate heat, for example, in animals. We've seen quite a bit about growth and development, particularly in plants, how plants use differential growth to respond to their environment, how primary and secondary growth occur. Response to the environment has been a primary theme for this latest section of the course, both in plants and animals, but also earlier in the course when we talked about cell signaling at the cellular level, how unicellular organisms respond to their environment. We've seen a little bit about reproduction in the synthesis of DNA and cell division. As we move on through biology, we'll see more of the details on reproduction in plants and animals. We've talked a great deal about regulation, homeostasis, negative feedback loops, and how organisms are able to maintain their body conditions in the face of fluctuations in conditions in the environment, which is one of the fundamental things that any biological system needs to do. And we've seen a great deal of the result of evolutionary adaptation in this tight coupling between structure and function. And so we've seen this at every level of organization, from the proteins whose shape determines what they're going to do and how they're going to function in a cell, to the structure level. We've seen this coordination between structure and function in different anatomical structures, and all the way up to the whole organism level, how organisms are adapted to their environment. And we've also talked quite a lot about emergent properties as you move from one level of organization to another. We talked a great deal about this transition from thinking at the molecular level to thinking at the cell level. So we saw that, for example, at the molecular level, understanding the structure of hydrogen and oxygen separately really doesn't allow you to predict the emergent properties of water molecules, what's going to happen when you put those molecules together. It's the interaction between the two that determines those emergent properties. And we've seen this over and over again, going up through the levels of organization. So understanding just the properties of ribosomes and the nucleus and the endomembrane system, the endoplasmic reticulum and Golgi apparatus separately is not going to really tell you much about how cells behave, because you have to see how all of these different parts interact together to produce a phenomenon like exocytosis. And then finally, we've come back over and over again through the course to this list of things that all living things that are alive do, and we've seen section by section how homeostasis is maintained, energy management, transmission of information through space and time, through time being from generation to generation, through space being through some sort of complex multicellular body, or between different organisms, responds to the environment. And we've seen the evidence of evolution in this relationship between structure and function. We've talked a little bit about mutations that allow evolution. But next semester in BioCore 12, you're really going to dig into the details and the nitty-gritty about how evolution by natural selection is another means by which order in biological systems and complexity in biological systems can increase through time, and the details of how exactly this happens.